Hello, my name is Malachi Griffith. I'm an associate professor at Washington University School of Medicine, where I co-direct a cancer genomics and bioinformatics lab along with my twin brother, Obi Griffith. I would like to start by thanking the organizers for inviting me to participate in this workshop. Today, I'm going to talk about bioinformatics and immunogenomics approaches for neoantigen target identification. A major focus of my lab's work is on understanding how T cells can recognize tumor cells by their unique genomic features. Our goal here is to really leverage the immune system's remarkable ability to identify and destroy cells infected with non-self or foreign elements. In cancer immunotherapy, instead of the foreign element here being a pathogen that has invaded a cell, it is the tumor cell itself. The reason this works is because tumors arise by acquiring somatic mutations in their genome, and these genomic alterations create altered proteins with unique peptide sequences that can be recognized as foreign. These altered proteins unique to the tumor are referred to as neoantigens. Depicted here is an example of the simplest form of neoantigen, a single nucleotide change leading to a single amino acid substitution. In normal cells, the wild-type peptide may be presented by MHC, but it will be recognized as self by the immune system and effectively ignored. In the tumor cells, if the altered version of the peptide is presented, it can lead to T cell recognition and ultimately tumor cell death. Two relatively new forms of cancer therapy that both rely on this principle are checkpoint inhibition therapies and personalized neoantigen vaccines. Additional therapies involving different strategies for bioengineering T cells that recognize neoantigens are also on the horizon. In checkpoint blockade therapy, arguably the most established of these therapies, small molecule or antibody drugs are used to inhibit signaling molecules that the tumor upregulates to avoid immune recognition. This effectively takes the brake off of the immune system so that it can more aggressively recognize antigens of the tumor. In personalized neoantigen vaccines, on the other hand, a set of specific peptides corresponding to neoantigens are identified, formulated into a vaccine, and delivered to the patient to stimulate an immune response against specific unique features of each patient's tumor. A major challenge in this field revolves around identifying neoantigens like the one depicted in the simple example I showed on the last slide. It all starts with a tumor antigen, in this case, a protein sequence that is unique to the tumor because it arises from somatic mutations acquired by that tumor. In the endogenous presentation pathway used by MHC class 1, this protein is cleaved into small peptides, transported into the endoplasmic reticulum, where it may be loaded onto an MHC molecule, and finally, this peptide MHC complex is trafficked to the cell surface, where it can be recognized by the receptor of a T cell. Since each person's immune system has an incredible diversity of T cell receptors, it is often assumed that if a neoantigen is robustly presented on the surface of the tumor cell, it can be recognized by at least one TCR. The remainder of what I will discuss is focused on how we predict the neoantigens unique to each person's tumor that can be presented and recognized in this fashion, and also how we can model the features of these neoantigens to predict their likelihood to ultimately be immunogenic and therapeutically relevant. Just a few years ago, doing this was nearly impossible, but now with advances in sequencing technology, we can rapidly identify neoantigens. We start with a sample of the tumor and match normal sample, and we isolate nucleic acids. Tumor and normal DNA are subjected to deep whole genome or exome sequencing, and tumor RNA is subjected to bulk RNA-seq or cDNA capture sequencing. We use exome sequencing of the DNA and cDNA capture sequencing of the RNA almost exclusively in our studies. These sequence data are then fed into an elaborate analysis pipeline whose goal is to identify, characterize, and prioritize neoantigen candidates, and then use these to develop a neoantigen vaccine personalized to the unique features of this patient's tumor. We implement our sequence analysis pipelines using the common workflow language, an open standard for bioinformatics workflow definitions. The workflow manages dependencies between steps in the pipeline and parallelization of the computational work. The actual computational tasks leverage Docker containers for every tool and step. The entirety of these pipelines, including all workflow and container specifications, are published under open source licenses using the version control system Git. These approaches collectively help to facilitate reproducibility and quality control as improvements are implemented in the pipeline, and portability so that the compute can be done efficiently on local or cloud-based compute clusters. We initially became interested in neoantigen identification after some early proof of principle studies where neoantigens were identified from a patient's somatic variants, used to create a personalized vaccine, given to a patient, and demonstrated some promising immune responses. Results from one of the first such studies conducted at WashU and reported in 2015 are summarized here. This figure shows pre and post-vaccination results depicted for 3 melanoma patients. The x-axis of each of these plots corresponds to a sort for CD8 positive T cells. The y-axis corresponds to a sort using MHC peptide dextramer. This pulls T cells specifically reactive to each patient's individual peptide MHC. So signal in the upper right quadrant of each plot represents T cell responses to a peptide included in the patient's vaccine. A total of 21 distinct peptides are depicted here, 7 for each patient. The red boxes depict a T cell response specific to a peptide used for vaccination where there was some evidence for these T cells before the vaccine was even administered. So in these cases, the vaccine seems to be increasing an existing immune response. The blue boxes depict detection of a T cell response specific to a peptide where no such response was detected prior to vaccination. So these indicate a new response stimulated by the vaccine. The thing that really caught our attention from this study was the significant difference between peptides that seemed to work and those that didn't. Even though all neoantigen candidates were carefully selected, only about 40% wound up working. So we became interested in trying to understand their molecular features and to hopefully improve these predictions and learn about how to create the optimal neoantigen vaccine. In general, neoantigen prediction remains a major biology and bioinformatics challenge. Shown here is a 3D depiction of a peptide MHC T cell receptor complex. These 3 components are forming the elaborate handshake required for recognition of an antigen by a T cell. Each of these components is incredibly complex. Starting at the bottom, there are more than 5,000 known HLA alleles that encode the MHC complexes. These genes are some of the most genetically diverse in the human genome. Luckily, there are some great resources like the EBI's IMGT to help organize these data. Next, there are more than 200 trillion possible short peptide sequences that can potentially fit into the groove of each MHC molecule, though only a tiny fraction of all peptides will be recognized by any one particular MHC allele. As I will discuss in a moment, we now have an impressive diversity of large-scale data sets relevant to peptide MHC binding and a growing number of constantly updating algorithms trained on these data. Finally, there is an almost infinite diversity of T cell receptor sequences that could recognize the peptide MHC complexes formed. Once again, we are lucky to have access to some remarkable recent advances in TCR repertoire profiling. And of course, these 3 major components, the MHC, peptide, and TCR, don't function in isolation, but rather as part of an elaborate cellular machinery and signaling network with many important factors both upstream and downstream of this complex. The vastness and complexity of this system and the challenge inherent in marshaling all the relevant data and algorithmic predictions is what really inspired us to work on the related bioinformatics approaches. Hopefully by this point, I've convinced you that identifying and characterizing neoantigens is an important challenge with therapeutic implications. Identifying neoantigens and designing a personalized neoantigen vaccine involves a complex workflow. At a high level, one starts with tumor samples from a patient. Sequence data are mined to detect a variety of somatic variant types and determine the HLA alleles of the patient. Additional predictors are applied to anticipate peptide processing, transport, and stability. The unique peptides of the patient's tumor are modeled to predict their binding and presentation by the patient's MHC alleles. Finally, a set of usually 5 to 30 of the best candidate neoantigens are selected and packaged into a vaccine and delivered to the patient. These vaccines can be formulated in a variety of ways, and this can influence the design process. All of this must be done as quickly as possible to be useful in a clinical setting. One of the exciting things about this approach is that it builds on everything we've been doing to advance tumor genome sequencing and analysis over the past years. Literally several dozen bioinformatics tools developed by groups around the world are needed to go from raw data to a finalized vaccine design personalized for each patient's tumor and MHC complement. And each of these tools must have carefully tuned parameters and optimized inputs, which in many cases require fine tuning for different cancer types, patient cohorts, and clinical sample characteristics. Of course, in order to create the optimal process for neoantigen modeling, we must think carefully about this question. What are the fundamental features of a good neoantigen? These are some of the high-level considerations that many in the field have been focused on. Arguably the most important characteristic, and certainly the feature that most clearly distinguishes neoantigen approaches from decades of past work on tumor-associated antigens or cancer testis antigens, is the uniqueness of the peptide sequence to the tumor. The major appeal of neoantigens is that they should never have been seen by the immune system prior to the development of the tumor. This is theoretically desirable from the perspective of avoiding immune tolerance and autoimmune responses. A good neoantigen is also ideally present in every tumor cell, though there is also the possibility that targeting subclonal neoantigens could still encourage a broader immune response. Since presentation of an antigen is key, expression is a requirement. There are some reports suggesting that the robustness of expression is even more critical for class 2 MHC presentation than it is for class 1. The strength and stability of binding of the peptide to the MHC groove is a major focus of all neoantigen modeling approaches. Finally, the choice of vaccine delivery platform may impose some limitations on neoantigens. For example, if using a synthetic long peptide or dendritic cell vaccine approach, the inability to synthesize some peptides may disqualify some candidates. One major area of focus in the field of neoantigen characterization is exploring the different possible sources of neoantigens and considering how different sources may influence the fundamental features of a good neoantigen described on the past slide. Shown here are just some of the major neoantigen sources, each generally requiring a highly customized workflow to accurately detect and annotate them. First, we have simple point mutations, an example of which I've already shown. Second, we have small deletions and insertions, which are of great interest because they can lead to frame shifts. Frame shifts may be undesirable in some ways. For example, they might be subject to nonsense-mediated decay and therefore have reduced expression. But if they are expressed, they have several interesting advantages. For one, they give rise to longer stretches of peptide sequence that are much more unique to the tumor genome than a peptide with a single amino acid substitution. Furthermore, in some cancer genes, many tumors can have frame shifts around the same position in the protein. We see this in P53, for example. If these lead into the same altered reading frames, this raises the possibility of neoantigen peptides that are shared across individuals. Aberrant tumor-specific alternative splicing can similarly lead to rich sources of neoantigens when again a reading frame is shifted by an event or a section of intron is retained that is not normally expressed. Interchromosomal translocations can give rise to fusion proteins that also potentially harbor highly specific neoantigen peptides. And in some cases, these are also strong drivers of tumor biology, which would be an advantage. And it just goes on from there. Neoantigens can arise from pseudogenes that have been reactivated, historical or contemporary viral integration events, read-through translation events, and so on. And once expressed, peptides can also be post-translationally modified by methylation, phosphorylation, acetylation, et cetera, to give rise to even more diversity. So identifying the neoantigen in the first place is practically itself a whole field of bioinformatics. But once identified, each is considered with respect to the fundamental features I previously outlined. All of these features, there has been a major focus on modeling of the peptide MHC binding relationship. This focus has arguably come at the expense of other factors that also matter to the ultimate immunogenicity of a candidate neoantigen, but there is no doubt that this is certainly a critical component. Here I am showing a three-dimensional model of an MHC molecule, in this case, the HLA-B0801 allele. This 3D model was generated from a crystallized version of the protein with a 9 amino acid peptide in the binding groove. The MHC protein is shown in green. It forms a complex with the B2M protein shown in blue. And the 9 amino acid peptide is depicted in pink. It is nice to visualize this complex in 3D like this, but there are very few such reference MHC structures binding only a small number of example peptides. Also, there are so many degrees of freedom within these peptides and their interaction within the MHC groove. In practice, we cannot model the peptide MHC interaction by a 3D in silico docking approach, as we might do for small molecules in more traditional drug development. However, there are now many competing algorithms that do model peptide MHC binding using different types of data with a variety of statistical and machine learning approaches. As with any model, it is only as good as the data it is trained on. So the data used to train peptide MHC predictors is important to think about, and it falls into two major categories. First, there are large in vitro binding assay data sets. These involve testing a set of MHC alleles against a library of many peptides using competitive binding assays. Essentially, you determine the concentration at which a test peptide outcompetes 50% of a labeled reference peptide. This gives you an IC50 value for each peptide tested, but it's not feasible to test every MHC allele or anything close to every possible peptide sequence. But the premise is that with data for enough MHC alleles and enough test peptides, you can extract features of those MHCs and peptides and create a generalized predictor for any MHC and peptide. The second major category of data involves peptide elution mass spec. In simple terms, this involves use of an antibody for a conserved portion of the MHC molecule. And then using cells expressing MHC, you pull down complexes with this antibody, and then you elute off the unknown peptides being bound by MHC, and you feed the L08 into a mass spec to identify the peptide sequences that were bound. As you can imagine, both of these types of data have their caveats. I could probably spend 30 minutes discussing those, but suffice it to say, many of the recent binding prediction algorithms seem to be combining both data types and coming up with reasonable solutions to this part of the problem. To give just one example of a very specific area for improved neoantigen predictions that we have tackled in my lab, consider the very simple neoantigen in the top left. The mutant version consists of a single amino acid substitution. For this type of neoantigen, the uniqueness factor is very important to consider. In particular, when the mutant and wild type are so subtly different, one must consider carefully how they might be presented and recognized by the immune system. Depicted here in cartoon form is an MHC peptide TCR complex. In these complexes, there are residues of the peptide that are critical to binding by the MHC molecule. These are called anchor residues and are shown on the bottom, interacting directly with part of the MHC group. The other non-anchor residues of the peptide are more critical to recognition by the TCR and are shown at the top, facing the TCR. The reason we care about this in the context of neoantigen presentation is that if the mutant amino acid is an MHC anchor residue and both the mutant and wild type peptides are strong binders, then the residues actually being shown to the immune system could be the same. This could lead to tolerance where the immune system does not recognize the peptide as foreign, meaning it would not be much use in directing a response against the tumor, or potentially worse, it could lead to autoimmunity where a response is stimulated that could impact healthy tissues. Conversely, if the mutant amino acid is in a TCR contact residue, it might not matter if the wild type version of the sequence is also a strong binder because the residues being presented to the immune system are unique. Most existing peptide MHC binding algorithms and neoantigen workflows do not sufficiently account for this mutant versus wild type comparison, so we have worked to improve methodology in this specific area. And we created PVAC tools to facilitate the process of end-to-end neoantigen analysis, including consideration of the features I've just introduced as examples and many more. PVAC tools is a highly modular analysis toolkit implemented in Python that is designed to support each step of neoantigen analysis. Its modularity is aimed at supporting customized approaches and use cases for neoantigen analysis ranging from basic to translational studies. We use it in several contexts, but probably most heavily to create personalized neoantigen vaccine designs in collaboration with an immunotherapy tumor board for a variety of clinical trials. I don't expect you to read all these details, but in broad terms, it breaks the process down into components of data QC, HLA typing, various kinds of somatic variant detection, prioritization, visualization, and packaging into a vaccine formulation. To maximize the broad adoption of PVAC tools and integration into other workflows, we have tried to always use standard file formats for inputs and outputs. Essentially, everything it needs for most functionality can be encoded in a single input VCF. In order to incorporate some features specific to neoantigens, we created another tool called VA Tools to help users prepare this VCF. If you want to learn more about this tool, I would encourage you to visit pvactools.org. Of course, my recommendation of PVAC tools is somewhat biased. If you're new to this area, I would also strongly recommend two other suites of tools that include a wealth of useful functionality for neoantigen characterization. In particular, IEDB is a very well-established and rich source of both data and tools for fundamental assessment of immunological characteristics. And a bit more specific to tumor biology, the OpenVax group has some really wonderful stuff as well. In general, you can learn a lot about the biological characteristics of neoantigens, the kinds of experimental data being leveraged, and the quantitative features being derived from these by examining some of the tools in this field. A very recent exemplar of this idea is NeoFox, a tool from Uyghur Sahin's group that is currently unpublished, but seems like it will be soon. Given a list of candidate peptides and other basic genomic characteristics, this tool will efficiently extract features for modeling neoantigens, and the features supported cover a broad swath of the currently understood important biological characteristics of a neoantigen, which are summarized briefly here. As I'm sure you can appreciate by now, there are a lot of relevant factors that we want to take into account when modeling neoantigens. In our own studies, we are tackling this challenge in two ways. The first involves trying to organize and synthesize important features into a report and developing an interactive graphical interface to aid our immunotherapy tumor board in evaluating each neoantigen and selecting candidates for cancer vaccines. Shown here is a brief demo of this interface, which we call PVAC View. This tool is designed to leverage the PVAC tool suite of tools. However, We use a simplified aggregated report of candidates and JSON formatted input for the features of each neoantigen in the hopes that others could plug it in to the end of their own established workflows. To review candidates, you start by loading your input files. You're then presented with a list of candidates summarized to the level of somatic variance and preliminarily tiered with one line per mutation. From this view, you can see the number of peptides meeting basic criteria and details on the variant itself, such as gene expression, allele-specific expression, tumor clonality, and MHC binding metrics for the best candidate arising from this mutation. The interface then allows you to drill down and get a lot more information about each individual peptide, including transcript-specific expression, annotations, and expression information, the peptide sequence, the position of the mutated amino acid relative to MHC anchor site predictions, and results from multiple individual MHC binding prediction algorithms. We currently use an ensemble approach with 8 to 10 algorithms used to evaluate every candidate. And as you go, you can select a status for each candidate, and when the review is complete, you can save and export your prioritized candidates. A second general strategy we are pursuing to deal with the complexity of factors to be considered is to develop more sophisticated predictors that will take them all into account. With this in mind, we've been collecting data from published studies to create a training data set. Our focus with these data has really been to identify neoantigens that have been subjected to validation experiments in humans to determine an actual immune response, not just MHC binding. We are particularly interested in data from patients where immunological assessment has been performed before and after vaccination. In addition to identifying and obtaining agreements to access the genomic data for all these data sets, we've been working to perform harmonized processing of these data, which are quite heterogeneous. We've also been working to design and extract features for a machine learning exercise and to develop a predictive model using a neural network approach. But the thing that is really lacking in this effort is the results from neoantigens actually evaluated in human patients. With that in mind, we have involved ourselves in several clinical trials that are testing personalized neoantigen vaccines, all of which have some detailed plan for immunomonitoring of the patients. At this early stage, these are mostly phase 1 or 2 trials, but nevertheless, they allow us to explore and refine our neoantigen analysis approaches in a variety of tumor types and clinical scenarios, and results are gradually starting to emerge from these trials. If we can eventually aggregate data across all the trials we're involved in, we could in theory have data from 150 patients, and assuming an average of 10 peptides per patient, a total of 1,500 vaccine peptides tested in a real world setting. With that, I will end here by acknowledging all the members of our lab and the PVAC Tools team in particular, who are highlighted in bold here. I would also like to acknowledge our wonderful collaborators, particularly for this work, Will Galanders, Chris Miller, Gavin Dunn, Beatrice Carino, Ravi Upaluri, and Todd Feniger, who leads some of the clinical trials we are most actively engaged with. And last but not least, our sources of funding. And for this work, we are particularly grateful to the NCI ITCR program. Thank you for your attention. I'll be happy to take any questions you may have.